my pleasure today to introduce Dr. Jocelyn Richard of the University of Minnesota, Department of Neuroscience. Thank you so much, Jocelyn, for agreeing to be interviewed today.
All right. Thanks for the invitation.
Yeah, so we're going to start our conversation today with Dr. Richard's recent paper entitled Alcohol Availability During Withdrawal Gates the Impact of Alcohol Vapor Exposure on Responses to Alcohol Cues, which was published this year, 2022, in Psychopharmacology. It's Carpio et al. So maybe we could start by asking you, Jocelyn, to tell us a little bit about what you did in the study and what you found and the main findings, that kind of thing.
Sure, happy to. So our main goal here was to look at how chronic intermittent exposure to ethanol vapor, which is a commonly used model of alcohol dependence, impacts behavioral responses to alcohol cues. Because a major focus of my lab is on sort of behavioral and neural mechanisms governing how cues can drive alcohol seeking and addiction-like behaviors. So in this study, we trained animals in a discriminative stimulus task with a cue that predicted alcohol availability. And then after animals had learned the cues, they experienced 3 weeks of intermittent exposure to ethanol vapor. Another important manipulation was that only half of the animals were able to access oral alcohol when they were in acute withdrawal. So each sort of cycle of ethanol vapor exposure involves 4 overnight 14-hour sessions of vapor exposure. And so some animals also had the opportunity to orally consume alcohol twice a week during that period of time. And I mention that because it had a big impact on how the vapor exposure affected cue responses. So basically, in animals that had the opportunity to consume alcohol when they were in acute withdrawal, we saw increases in how reactive these rats were to the alcohol cues. And in animals that didn't get this access, we actually saw a slight decrease in their reactivity to the cues. And so just the physical exposure to alcohol alone to induce dependence wasn't sufficient to drive increased cue reactivity. The animals sort of, we think, had to have the opportunity to learn that maybe alcohol had a new value after this vapor exposure.
And that's because it basically makes them not be in withdrawal anymore. They feel better basically when they get the drug. Is that right?
Right. Presumably that's at least our interpretation, is that during these sessions, the alcohol is alleviating some of their withdrawal-related symptoms, and then that appears to transfer later to the alcohol cue, even though they never actually are presented with the cue and the alcohol together when they're in acute withdrawal.
I see. That's very interesting. So it kind of brings up the the idea of dependence, which I assume is what you're inducing with the ethanol exposure at those sort of high levels for long periods of time in a given day. They're becoming physically dependent, right, on the ethanol, and then perhaps learning that alcohol can make you feel better after that experience. Like, we're studying dependence essentially. And I'm wondering if you have any comments on whether how dependence plays into the broader phenomenon, perhaps of addiction. One of the symptoms that contributes to a DSM diagnosis, but it's one of them. Could you say anything about that?
Right. And that's a very important question, and some of what sort of drove the desire to look at this model of dependence in the context of cue-elicited responding, which we think is very important for addiction. And dependence gets a lot more focus in the alcohol field than I think it does in sort of the non-alcohol addiction research field, so all of the other drugs, which is interesting. And part of that is, I think, because alcohol dependence can produce strong withdrawal symptoms and can produce problems that are not actually driven by addiction per se, but are related to this phenomenon of physical dependence. And so I think what this suggests is that certainly physical dependence and increases in addiction-like behavior can go together in some cases, but they don't always go together. And there are certainly drugs that can produce physical dependence that don't produce addiction-like behaviors. And so understanding mechanisms of dependence are not always going to tell us anything about mechanisms of addiction specifically.
Although, as your paper shows, when you're in with physical dependence, I guess, and then withdrawal, then that can affect your learning about cues and things, which then may be potentially related to, relapse, for example, and addiction. Is that fair to say?
I think that's fair to say. Under certain circumstances, symptoms related to dependence can interact with other factors that influence addiction to drive, like alcohol use and alcohol-seeking behaviors. But we also can see increases in those addiction-like behaviors without these levels of extreme alcohol exposure or these patterns of alcohol exposure. So in the paper, the figures are set up to compare, controls and vapor-exposed animals that had this access to oral alcohol and controls and vapor-exposed animals that didn't. But you could also imagine a graph where you had the two different types of controls next to each other, and the controls that didn't get access to oral alcohol during this 3-week period, so they never got any alcohol during this period, their cue responding is very similar to the vapor exposed animals that got that access, and it's higher than the controls that continued to get access to alcohol twice a week. So one question is that related to abstinence from alcohol, and is the absence of alcohol exposure also equally capable of driving addiction-like behaviors, which is what seems to be the case, at least in this data set.
I see. Interesting. So there's lots of things going on here that are interacting for sure. And then with regard to dependence, would you say that just from a kind of conceptual perspective that it plays a different role in addiction to ethanol or alcohol than it does in, let's say, cocaine addiction or some other drug of abuse?
I think that that's very likely in sort of the short term, in terms like when people and non-human animals are engaged in sort of active drug or alcohol use, that dependence may play more a role in the sort of the amount of alcohol consumption or escalation. And clearly we can see effects, rats and mice will drink more, especially in the period of time where they're being exposed to alcohol vapor. But in the long term, when you think about sort of long-term risk of relapse and the chronic nature of addiction, it's not clear that dependence has a stronger effect on like long-term risk of relapse for alcohol than it does for other drugs, and that it's likely non-dependence related mechanisms coming into play at that point, or at least that's my interpretation of the literature.
Yeah, no, that's kind of how I think about it as well. Then just out of curiosity, what's your take on sort of those long-term changes that may be at the core of the disorder, chronic relapsing disorder of addiction? Would you guess that like those are similar across drug classes? And I think there's some evidence that, like, for example, if you're studying relapse, like some people have found, Yubin Shahan's lab and others have found that, like, even the cue responses and things that occur, that presumably don't have much to do with dependence itself, may have different neural mechanisms. So do you have a sense that there's like one thing that is addiction in the brain we're looking for, no matter what drug we're talking about, or even maybe gambling and sex and other kinds of things, or is it more complicated than that?
I mean, I think it's more complicated than that. That's the easy part of the answer. It's more complicated. I also imagine there are some shared mechanisms, but that even within a drug class, that there's going to be differences based on the type or pattern of use, and also potentially differences between species or strains. we see some sex differences here in this study, more on measures related to that I think are related to dependence than to sort of motivation and addiction-like behavior. But even when you're considering, animals of the same strain that differ on some factor like sex, you can still get different mechanisms for the same ultimate behavior, which suggests that it's very unlikely that there's going to be one mechanism across all classes of addictive drugs and all people with all different types of problematic use.
Yeah, that makes a lot of sense. Yeah, and then another thing that kind of that you just mentioned as a broader issue, I know you found quite a few sex differences in this paper, and your lab and my lab and many other labs have been finding sex differences in practically everywhere you look. Could you speak a little bit more about sex differences in the brain, either in the context of your data here or just more broadly, whatever you kind of talking about?
Right. So the most consistent sex difference that we tend to see across all of our alcohol studies, which is consistent with a lot of other people's work, is that the females, even without these kinds of sort of vapor exposure manipulations, consume more grams per kilogram alcohol across a variety of models. And so in thinking about some of the sex differences we report here, which include, so we only see vapor-induced increases in alcohol consumption in males, and that replicates prior reports. And we only see vapor-induced increases in aversion-resistant drinking in males. So one possibility is that the females are sort of starting out more motivated for alcohol or displaying more addiction-like behavior without sort of excessive alcohol exposure, and that this vapor exposure is making the males more like females, and in which case the question is sort of what is the existing neural mechanism that is making the females initially more vulnerable. But at this point, it's hard to say if it's really that the males are more sensitive to the vapor exposure, or that the females just don't need the vapor exposure to reach that same level of behavior. And I think the answers would be different depending on your interpretation of that.
Yeah, these are certainly very complex issues. I mean, one thing that always just sort of strikes me, because we just constantly find sex differences in, again, practically everywhere we look, except for ventral pallidum, by the way. In those studies, we have not found sex differences for whatever reason, which I bring up because we both are interested in that structure as well. It just strikes me as just very strange, because men and women, when it comes to most basic features of behavior, are similar, or at least equivalent in most cases. Perhaps there's some population differences on certain types of things, but a lot of the stuff we're studying and finding sex differences in the brain on, there's no obvious sex differences in behavior. And that just strikes me as weird from an evolutionary perspective, that evolution would have sort of two solutions to this problem, at least two solutions, or probably lots more than that actually. Do you have anything, I'm just curious what your take on this kind of thing is. Why are there such pronounced sex differences everywhere we look?
Right. So I guess first I would say that when we talk about sex differences, I think it's important to keep in mind that those sex differences can vary a lot across different strains of rodents, and I basically never really want to generalize from sex differences that I see in Long Evans rats to saying anything about sex differences in general across other strains or species, etc. And that a lot of the time when we see these average sex differences and say the males are expressing more of this effect than the females, I want to be careful not to make too much of those average sex differences, because there's still overlapping distributions. And so I think we have to be careful of saying, oh, this is not a mechanism that ever happens in female rats or that always happens in male rats. And I think one example, there was a recent paper from David Yench's lab where they looked across lots of different strains of mice, and there wasn't one kind of sex difference that they found. And so I think some of this is that if we're sampling across animals with more biological variability, then we're going to see some differences popping up. And that's not necessarily going to like tell us about differences between like human men and women. But I feel like I've strayed a little bit away from your question.
No, I think that's a great answer. It sort of is a little, in some ways, potentially discouraging, if that in fact all the stuff we're finding is like really, it really matters what kind of rat you're talking about, let alone like, you know, does it have anything to do with humans? And if these things are so variable even across different, you know, bred populations of rodents, you would easily imagine that they might be different across species as well. I don't know if that if that's necessarily discouraging, in that we should understand how these work in any given species, I think is a valuable goal in itself in terms of defining principles that then might apply in perhaps different ways, but via principles also in human brains.
Right. I would say I don't find it necessarily discouraging. I think it can, like, looking when we see sex differences like this, it can help us to sort of refine the models we're using and maybe try to make them more relevant to like more animals, including humans. And so I think this kind of moves us in the right direction. And that I also think that when we're trying to understand these complex behaviors, fully understanding one type of complex behavior within like one strain of a particular animal is still useful, because at least it would tell us that we would give us a framework for understanding behavior across other strains. Because I don't think we've gotten to that point for any behavior in any strain of animals, which just means there's a lot to do. But yeah, but in this particular case, so with the aversion resistant drinking, vapor exposure had been shown to increase aversion resistant drinking in male rats before, but just those studies didn't include females. Here we're showing that the vapor exposure doesn't increase aversion resistant drinking in females, but the controls are pretty aversion resistant to begin with. And if the goal is to understand just like continued alcohol use despite like aversive outcomes, then that might just suggest maybe vapor exposure isn't the best model if we want to understand this mechanism, or we have to use multiple models and look at individual animal behavior, and not just looking at the effects of vapor exposure alone isn't like the path forward in understanding aversion resistant drinking, for instance.
Yeah, that's certainly fair enough. Yeah, then it kind of transitions into another question I had that you brought up, is this issue of just individual differences in general. Of course, there's sex differences, but then there's also individual differences that are quite considerable. And I noticed that quite a few of the animals in the study didn't voluntarily drink at all. And so you didn't look at them because you're looking at drinking animals. But I'm just curious if you can comment a little bit about the role of individual differences and our understanding of addiction and how we treat this in animal models and things along those lines.
Yeah, I think that's a great question. And as you point out, in this study, about 25% of rats were excluded after the initial home cage exposure stage. Not because it wouldn't be interesting to understand how vapor exposure affects how those animals respond to alcohol cues, but just that animals that drink below a certain level tend to not really learn this task. And so we didn't do that in the second experiment in this study. We were looking at vapor exposure prior to cue learning, where we didn't really see where we saw, if anything, decreases in alcohol seeking in the task after when they were trained after alcohol exposure. And so like those rats themselves are very interesting. And we've seen lots of individual differences in the aversion resistant drinking as well, which I think can be useful as a tool for looking at neural mechanisms, along with these sort of exposure paradigms that can maybe push animals in one direction or another, but don't totally determine whether they're like more or less aversion resistant. It certainly makes things more complicated, but I think it makes sense that there are these big individual differences, just given like differences we see in how humans use alcohol and other drugs.
Absolutely. Most people, of course, drink and are not addicted to alcohol. And so that variability there, what's different about the people that are. And sometimes people say things like, 20% of my rats or whatever, my mice are addicted, or sort of like, these are the ones that will get addicted, and other ones are sort of maybe resilient to drugs or something like that, to becoming addicted once they expose to drugs. Do you think of it that way? Do you think that there's sort of like subpopulations within our outbred rat stocks that are just like little, just waiting to get addicted, and others that are just will never basically get addicted, or is there something else going on?
Well, I mean, I think I tend to think of it more as like risk or behavior along a spectrum, and less about sort of cutoffs for addiction like behavior or not. Though I think those approaches can be very useful in their own way. It's just not a kind of approach that I've taken. And so the rats in this study, presumably some of them started off with some sort of higher risk of responding to the alcohol in a certain way. But I also think it really matters, like the behavioral paradigm and like type of alcohol exposure really matters. And so I think a high risk rat isn't going to be a high risk rat in every model. And it's also going to include a lot of factors that are really hard for us to quantify in terms of housing and background. And so I think there's very important work being done on factors that maybe confer risk. I'm more interested at sort of the later stages, and like once you see the behavior, what are the neural mechanisms that are driving that?
Totally. Yeah, that makes a ton of sense. And I think it kind of like, an implication I think of your point is that there may not be such a thing as like risk of addiction. It might be, for example, sensitivity to punishment is one factor that you can individually vary on and may be relevant to addiction. Another factor could be susceptibility to respond to cues in general, or effects of being dependent on the drug. Any of the things could individually differ and lead to what seems to be a addicted phenotype, but in fact it occurred through different sorts of risk in the first place.
Yeah, definitely. The sort of, I don't think it makes sense to talk too broadly about individual risk for addiction as opposed to like individual risk for certain behaviors associated with addiction or certain types of issues. But
totally. Okay, and then so this one other thing I wanted to ask you is, so you're the first person that's like largely an alcohol researcher that I've interviewed for this series, and I've had the impression as a person who's not studied alcohol before that there's a little bit of almost I don't know if it's quite a divide, or but at least sort of separate groups of people that are studying alcohol versus lots of other drugs. And obviously this is imprecise and it's not entirely true, but I have a sense there's something like that. And part of this is there's alcohol specific conferences and things along those lines. But I'm curious if you get a sense that there's that kind of line in the field in some ways, and if there's what you have, if there's anything interesting to say about that kind of thing, if it's helping us, hurting us, the reason for it, good reason, bad reason, etc.
Right. I yeah, it is interesting sort of being in the alcohol research field, but also being someone that's generally interested in addiction beyond alcohol, and also a lot of what my lab does is also doesn't involve alcohol at all and is focused on sort of basic neural mechanisms of reward and motivation, and sort of straddling those two fields a bit has been interesting, because there are different priorities, some of which is driven by the funding priorities of the main institutes, so NIDA versus NIAAA. And it does, and there are reasons for that separation, but in general, it'd be useful to see for there to be more crosstalk, because alcohol is special in some ways, but probably not in other ways. And it would be nice for it to be easier to get funding to look at shared mechanisms for like cue-elicited alcohol seeking and cue-elicited seeking of other drugs. And so there's definitely a limitation of the current system.
Yeah, that makes sense. Obviously, one of the things that's also different about alcohol compared to the other drugs is that like it's vastly more used than the other drugs, and it's in terms of harm to people in the U.S., for example, it's going to be way up there, if not at the very top, just given how common it is and how socially accepted it is and stuff like that, which obviously probably has to do with the funding structure and whatnot as well. But yeah, these are interesting issues. I'm glad to hear you calling for additional overlap. I'd like to see that too, because there may be things that are common, maybe things that are different, and one way to find out is to do research. So people like you, I think, can really push that sort of question forward. Great. Well, this has been a really interesting conversation, Dr. Richard. Thank you so much for taking the time, and good luck with your future studies on this topic. I look forward to hearing more.
Yeah, thanks so much for inviting me. It was great.